When you're learning about cell division, one of the trickiest things can be getting down what all these C and K words are. So we're going to look at this in a couple of different ways. First of all, when you're looking at DNA, it exists in a stringy form and a condensed form, but both of those forms in the nucleus are chromatin. Chromatin is an arrangement of nucleosomes, so you've got these histone beads and then the DNA wraps around them like string. And that's how DNA exists in the nucleus, is with this protein, histones primarily, but lots of different sorts of protein, condensed into this fiber we call chromatin. So both chromosomes and chromatids are made of chromatin. So what the heck is the difference between a chromosome and a chromatid? Well, it's all about timing. In G1, you have a single long stringy chromosome every time we talk about chromosomes. So we have 46 of these. During S phase, that chromosome or those chromosomes are replicated. So for every one chromosome you had, now you still have one chromosome, but you have two sister chromatids, and they're held together by a series of proteins called cohesin, and so that keeps everything organized. It's long and stringy. You want to keep those fibers together after they've been replicated. And so that's what the DNA is going to look like during G2 as well. You've got one chromosome and two sister chromatids for every one chromosome. During mitosis, that fiber condenses, and so this is when you start to see that compact rod-like DNA. It's already been replicated, so it looks like an S. The cohesins only hang on at the centromere region, and so a lot of them have been broken down. And so you see that X shape. And so that starts during prophase and lasts until anaphase when the cohesins, the rest of them, break down and those chromatids are split apart. And so now they are both known as two chromosomes. So at the beginning of N phase, for every chromosome you still had two sister chromatids. Now during anaphase we start to call them chromosomes again. There are no chromatids anymore as far as what we're going to talk about. So just zooming in on that replicated chromosome at the beginning of mitosis, the whole thing is a chromosome. The sister chromatids are the two independent fibers made of chromatin, that mustard yellow color. The cohesins are the proteins holding these sister chromatids together. The kinetochore is a bunch of proteins that gather on the outside of those sister chromatids on opposing sides. And that whole region where those cohesins and kinetochore proteins bind, it's, slightly different in sequence and folding and condensing than the rest of the chromosome, and we call that the centromere region. And so it's just a region of DNA, just like the telomeres are regions of DNA. So that, in a nutshell, are those pesky C and K words you need to know when you're talking about chromosomes and cell division.